we'll greet Dr. Niema Moshiri, Assistant Teaching Professor of Computer Science and Engineering at UCSD with a focus in computational biology. Take it away, Niema.
Awesome, thank you. So I'm gonna do the obligatory sharing slides and hopefully people can see my slides.
It's good.
Yeah, perfect, awesome. So yeah, thanks for the introduction. I'm glad to be here. So as you mentioned, I'm at UC San Diego and my research area is in bioinformatics. And specifically during the COVID-19 pandemic, I've been working with some folks at UCSD to try to accelerate some of the real-time analyses that we want to be able to do kind of on a large scale. So in this talk, I'm going to give an overview of just kind of all of the general background that goes into the COVID-19 sequence data analysis. And then I'm going to transition to some information about how we've leveraged cloud computing to help us with this. And I mean, generally, so I'm going to leave a little bit of time for Q&A just because I think maybe a lot of people here might be interested in the applications of this, but might not know some of the background. So I'll mainly focus on the background and the Q&A, and I'll kind of just briefly mention our results because they're not super novel in terms of cloud compute utilization, but it's just an interesting problem area that maybe people here would be interested in. But yeah, so here's a broad outline of today's talk. And first, I just want to give an overview of just what is viral molecular epidemiology. So public health, just to do some introductory definitions, public health is the science of protecting and improving the health of people and their communities. And generally, a standard public health approach for responding to some problem is the following. You have the problem and you have the response. And generally, you start with surveillance. So first, just trying to identify and characterize what exactly is the problem that's at hand. This is followed by risk factor identification. So now that we've identified the problem, what is the actual cause? And then intervention evaluation. So now that we have an idea of what causes this problem, what interventions actually work? How can we evaluate how good those interventions are? And then finally, we have implementation. So now that we know which interventions might be good, how do we actually do them? How do we implement those interventions? And that is the response. So some definitions about the field of epidemiology. So epidemiology is the study of the distribution and determinants of health-related states among specified populations. And it's the application of that study to the control of health problems. So kind of one central theme is how do we utilize epidemiology within public health practice? So a kind of general a structure for this is first discovering the agent, host, and environmental factors that affect health, determining the relative importance of causes of illness, disability, and death, identifying those segments of the population that have the greatest risk from specific causes of ill health, and then evaluating the effectiveness of health programs and services in improving population health. So that's kind of my like brief couple minutes introduction to what is public health and epidemiology. With those general definitions in mind, I now also want to give some background with regard to some evolutionary theory, and then we're going to later merge those two. So on the evolution side, I kind of want to introduce the concept of a phylogeny. So a phylogeny, it's just a fancy word for evolutionary tree, which people have probably seen at some point in their careers. Basically, we have the leaves that represent present-day species, and we have ancestral nodes that we typically represent as extinct, or we assume that are generally extinct. And the lengths of the branches in this evolutionary tree generally denote some form of evolutionary distance. So it could be some amount of like human time. So it could be years or seconds or generations, or it could be something that kind of translates into time. So in the area of phylogenetics, a lot of times we deal with number of mutations or some type of normalized number of mutations, because more mutations is more time generally. So you've probably seen phylogenies in the context of species, but it turns out that you can actually use this type of logic within the same species. So if I have a bunch of samples from individuals of the same species, I can use the same type of logic. So here I've drawn them as HIV samples. You can plug in whatever pathogen you're interested in, where the leaves represent individual samples from different people. And we can construct an evolutionary history that represents the relationships between those. Okay, so an epidemic, we can think of it as this kind of trajectory, the spread of a pathogen through a given community. And we can kind of model some of the aspects of this spread in a more kind of mathematical framework. So we can think of what's called a contact network, which is a graph, generally undirected, which nodes represent individuals and edges represent risky contacts. And by risky contact, I mean some type of interaction across which my pathogen of interest can spread. And this differs from pathogen to pathogen. So if I'm dealing with HIV, this might be like sexual contact. If I'm dealing with COVID-19, this could be maybe being in the same room together. Different pathogens will have different risky contacts. And then under this contact network, I have a directed subgraph, which I will denote the transmission network. So in this, the nodes are the same as in the contact network, but now my edges are specific transmission events, with each transmission event labeled with a time. So here, for example, this transmission event is from this person to this person at some time T1. So in the case of HIV, we generally assume it's in tree. In the case of COVID, we used to assume it's a tree because we didn't really have reinfection events, but now we're starting to have a couple, we're starting to have more and more reinfection events. So you might not necessarily assume that this is a tree. But for simplicity, for a short-term kind of localized epidemic, assuming that no one gets reinfected multiple times, you can generally assume it's a tree. Okay, so let's zoom in a bit, and then hopefully we can tie in our concepts of evolutionary trees into this underlying transmission network. So if we take one of the transmission events and just kind of zoom in on it, within the host individual, so the source, we have a bunch of viral lineages within their body. So the virus is just continuously evolving within the body of the host. So at the time of infection, there was some ancestral viral sequence that initially infected this person. And then, yeah, since then, it's kind of spawned off and branched off into this big evolutionary history within their body. At the time of transmission, we basically select one viral lineage that then gets transmitted to the recipient. So here I'm labeling this transmission event with the time of that transmission, and one lineage is being sent from the source to the recipient in what we call a bottleneck event. So just kind of like one lineage is the one that survives. And then after that, we have evolution continuing in both the source and the recipient. So here, the source, like the evolutionary history of the lineages, continued to evolve. And here in the recipient, that single branch has now started splitting into more and more branches. So we're getting a new lineage, or like a new set of lineages within the recipient. So let's imagine that these two individuals come into a clinic, and then, if it's COVID-19, maybe we do a nasal swab and then we get some samples from them, maybe periodically over time if we want, and then we sequence those samples. What we're effectively doing is sampling a few random lineages, maybe just one per person, maybe a couple per person, but we're randomly sampling a handful of lineages from these individuals. And we're not actually able to observe the full tree because all of these other lineages that we didn't happen to sample when we did our sequencing, we don't get to observe those. So essentially what we're actually observing when we do some type of a sequencing experiment is some reflection of this observed constrained evolutionary history, right? So you can imagine we had some complete evolutionary history across these two individuals, but if we just picked a couple leaves and then pruned any branches that weren't along the paths from those leaves to the root, that resulting tree is what we actually get to observe. So this is kind of like the true evolutionary history that we observe. And then likewise, we can think of evolution of the actual genome sequences as some evolutionary process over time, where time in an evolutionary tree goes down for forward in time, where we have some ancestral sequence that's the common ancestor of all of the viral sequences that I've observed. And then over time, along the branches of this evolutionary history, mutations occurred that then get cascaded down and get inherited in any descendants of where they occurred. So here, if I have like a mutation happen on this branch, all descendants of that branch are going to have that mutation inherited. Okay, so all this context, we can kind of put it together in some form of a generative process. So from a mathematical modeling standpoint, we can think of this entire process as basically, we have a contact network which we can model using any number of network models. So for example, the Erdo-Trendy model, the Barbasi-Albert model, a lot of random network models. So the contact network is kind of like a sampling under some network model, not really conditioned on anything other than the parameters. And then we can think of the transmission network as, well, okay, it has to spread across the contact network. So under any number of epidemic models, so any number of like compartmental or agent-based models, we can think of the transmission network as being some random process that's conditioned upon the contact network. So some random process given the contact network. And then similarly, these transmission events strongly dictate constraints upon our possible tree evolutionary histories. So we can think of the viral phylogeny, the viral evolutionary tree, similarly as a random process that's conditioned upon the given transmission network that we have. And then the evolution of the sequences we can think of as a random process conditioned upon the evolutionary tree that we're given, as well as, the root sequence, like the ancestral sequence, and whatever models of evolution that we have, like the model parameters. So this we can think of as the full start to end generative process, but in practice, we don't get to see any of those earlier steps. They're all hidden from us. In practice, all we get to observe are the viral sequences. And we want to somehow use some form of inference to get some insights about those prior steps, because those prior steps, especially the transmission network and the contact network, that's really where the public health intervention, the actionable information, is contained. So in this talk, I'm just kind of introducing the context of this entire pipeline, but I'm not actually going to be talking about the individual steps of these inferences backwards. I'm only going to focus today's talk on just this single step of how do we even get viral sequences from a bunch of samples. And if you're ever interested, feel free to reach out to me and I can give some information about these other steps as well. But yeah, for today, just given a bunch of samples, how do we reconstruct individual viral sequences from each of those samples? Okay, so now I want to talk about how does viral genome sequencing in the midst of a pandemic work? So first, just some background of genome sequencing. An analogy that I like to use is what's called the newspaper problem. So imagine that I have a stack of 100 identical copies of some newspaper. So each of those individual copies is identical. And imagine that I put a stack of dynamite onto those, right under those newspapers, and I light the fuse. So it's, of course, a hypothetical example. Don't do this at home. And then I explode all of those newspapers. And what's left are all these tiny charred fragments of the newspapers. And the computational problem here is using those tiny charred fragments, can I somehow reconstruct what the original newspaper said? And this is an analogy that we like to use for genome assembly. So if you think about it, you had a bunch of copies of the same exact newspaper, and you want to try to use overlaps or similarities that kind of can be overlain upon each other to piece together your genome. So the exploding newspapers problem is a good analogy for genome sequencing in that, well, a lot of those pieces probably just got completely exploded. So there might be some portions of the newspaper that we can't reconstruct just because we lost it. So in genome sequencing, there might be pieces of the genome that we just don't get to see. And likewise, we have multiple copies of the same kind of overlapped components of the sequence. Just like here, we have overlapped components, like repeats of the exact same fragment of the newspaper. So you're probably wondering, how is this relevant to sequencing the SARS-CoV-2 genome, SARS-CoV-2 being the virus that causes COVID-19? So biologists lack the technology to just take some viral sample and read the nucleotides of its genome from beginning to end, like how you would read a book. Instead, the way that we have these technologies work is we can basically get multiple copies of a given genome and then just break it up randomly all across the genome. So here we had a bunch of copies of ideally the exact same genome sequence, and we just randomly fragment it across the genome. And the technologies are able to sequence these short fragments, which we call reads. And really, we only get to see a small number of those fragments. Many of them are just lost based on random sampling error. And we want to use these small overlapping fragments to be able to reconstruct the genome sequence. So if we don't know anything about the genome sequence that we're trying to reconstruct. We call this the de novo genome assembly problem. We're constructing it from scratch. So in the midst of a pandemic, though, we're actually a little bit more fortunate. So I want to introduce this concept known as a reference genome. So a reference genome is a high confidence assembled genome sequence that is constructed as a representative example of an individual organism. So getting the first genome sequence is kind of difficult. You have to like really sequence deeply and get a lot of sequences to hopefully be as confident as you can that this really is a high quality genome sequence. And then once you have that, we can call that a reference genome. So the earliest reference genome that was sequenced was the human genome in 2001, which actually has recently gone through some major revisions. But since then, we've been sequencing a lot more genomes over time from different organisms. And for SARS-CoV-2, actually very quickly into the pandemic, so on February 3rd, 2020, less than 1 month after the World Health Organization had announced a mysterious coronavirus-related pneumonia in Wuhan, the first draft of the SARS-CoV-2 reference genome was published. And this was quite the historic feat, because this is really fast. Okay, so now let's put ourselves in the shoes of the people that were researching in the midst of the pandemic. What we want to be able to do is basically leverage that reference genome to help make the computation a little bit easier. So in de novo genome assembly, you basically try to infer relationships between reads purely from overlaps. So in other words, given all of these reads, these short fragments, you're kind of doing pairwise comparisons essentially in some way to figure out how they might overlap with one another. But what if instead, if we have this reference genome at our disposal, what if we instead compare the reads against the reference genome? So each of these reads, I can basically see where does it best match in my reference genome. So maybe this sequence matches here, this sequence matches here, this sequence matches here, so on. We call this process read mapping. And this is a very well-studied problem in bioinformatics. I can share my slides later if people want to get these links. But there's a lot of different tools that have been used to do this read mapping. And the one that I recommend for now is called Minimap2. That's what we're using in our pipeline right now. Basically, given a bunch of fragments and the reference genome, it finds where they each match against the reference genome. So let's say that I get my raw data, I run Minimap2, and I get all of these mappings of where my fragments match up the best against the reference genome. I can essentially just take a sliding window approach to reconstruct what we call a consensus genome sequence from a given sample pretty trivially. I can basically just start at the very beginning of my reference genome and see, out of all the fragments that happen to overlap with that position, what was the most frequent letter that appeared? And then I just kind of keep going position by position by position until I reach the end. And now for each position of my reference genome, I have found the most frequent letter that appeared in my specific sample that I have. And sometimes you might have a mixture, or sometimes maybe from sequencing errors, you don't necessarily know what the exact letter is. We also have ways of putting an ambiguous answer, like maybe I'm going to put N because I know there's some letter here, but I don't know what it is. But either way, we reconstruct a genome sequence using this reference. We can also use what's called amplicon sequencing, where we can simplify not only the computational aspect, as we just described, but also the biological sequencing aspect. So in a sequencing experiment, instead of just kind of blindly sequencing whatever we have in a given sample, we can actually design what are called amplicons, which are kind of like these targeted little sequencing like prefixes that help guide the sequencing experiment that target specific regions of the genome. So instead of just kind of like randomly chopping up the genome and sequencing little chunks, I can actually design targets that are spaced evenly across the genome that then lets me improve my chances of actually having at least a few reads that cover every single position of the genome. And then prior to actually mapping these reads, I have to cut off these like primers, these targets that I've created, because it's kind of like, it's essentially, I've designed the beginning of the sequences that I'm going to obtain to have a better mapping. So I have to trim those off to not kind of give false information. And I also have to trim low quality bases, so letters that I might not be familiar about. Don't worry too much about the specifics of this. Just trust that this trimming step, cutting off things that I can't trust, is a well-studied problem as well, and tools exist to do this specifically for viral amplicon sequencing. So you can just kind of leverage existing tools that are considered best practice. So now getting to the portion that I think is the most relevant to people that are attending this, now that you have the relevant context. A standard consensus sequence bioinformatics pipeline for viruses looks something like this, where we generally have these main stages of read trimming. So as I mentioned, cutting off the ends of our reads to throw away anything that might skew the results. We have read mapping. So given my trimmed reads that only contain the high confidence portion of those reads, actually finding where they match in the reference genome. And we have variant calling, which for each position of my genome, what letters do I actually think appear here? And especially I'm interested in what letters differ for my reference sequence. And then we have all these different optional analyses. So there's a bunch of tools that might try to like do lineage assignments. So if you've seen in the COVID-19 pandemic, you've probably heard of like the UK variant, or like B117, or these types of lineages. There's tools that you can do for specific viruses. So for COVID-19, there's a tool called Pangolin that then, given a sequence, will try to match it to what lineage it seems most likely to be a part of. Okay, so let's imagine that I'm running a massive sequencing course. So at UCSD, we're sequencing up to thousands of samples where each sample comes from one person. We're trying to do thousands of samples per day. So because of just how the sequencing experiment gets set up, the computational analysis runtimes are pretty heterogeneous across the samples. So here's the distribution of total processing runtime of a standard consensus sequence pipeline that we have implemented across all the samples that we have. And you see that some sequences finish, pretty quickly, whereas some of them take maybe even orders of magnitude longer than others. So in this distribution, the average is 13 minutes and the median is 14 minutes with a standard deviation of 3 minutes. And you might be thinking, whoa, those are tiny numbers. Like, why is this guy here talking to us about such small compute times when we're worried about scalability? So yeah, where is the computational bottleneck here? I just said one sample takes 13.5 minutes to analyze on average. That's not too bad. But I don't want to just sample one sample. I don't want to just analyze one sample. I ideally want to sequence every single sample that has ever been tested. So rather than just doing a nasal swab and then doing RT-PCR COVID tests or the rapid tests, I want to be able to actually sequence every single person whose sample I collect. So COVID-19 testing, it can tell you, yes, you probably have COVID-19, or no, you probably don't have COVID-19, but it doesn't tell you what lineage or what mutations or where did you likely get it from. And those are the questions that are really interesting to public health people. So at its peak, we had roughly 30,000 COVID tests per day in San Diego, which would take roughly 6,750 CPU hours per day to keep up with that. Again, it's not too crazy for the types of orders of magnitude that you've probably seen in this group, but it's something that it starts making it a little less trivial to set up a good infrastructure. So your first question might be, why don't you just build a massive high performance computing cluster? So I could theoretically I can execute each sample completely independently. So, if I have 6,750 CPU hours and I have 24 hours in a given day to keep up with it, that's like 282 CPUs running in parallel. That's not too bad. But the challenge is that the time to results is critical. So I cannot use a shared queued cluster. I would need something custom just for this purpose. And also there's generally somewhat of a knowledge gap in high performance computing usage by public health officials or biologists. So folks here might be very comfortable knowing how to use like Slurm or QSub or something like that and getting access to high performance computing. But the people that are generating these data sets and need those answers don't know how to do those types of analyses themselves. And another key challenge is that you have extreme heterogeneity between days. So even if I hire some folks that can build this system just for my purposes and run it for me, some days I have a lot of samples, some days I have very little samples, and I don't really want to just have this thing idling and waste money when it's not being used. And it's not really trivial to expand my compute resources in the future if I ever need. So maybe today's testing is some scalability, but maybe in a week, some breakthrough happens that we can now sequence a lot faster. It's not very easy for me to expand in the future if I need. So I'm going to introduce a pipeline that I helped develop called reflow, which leverages cloud computing to help this out a little bit. So reflow, you can find it on GitHub. It implements a standard viral consensus sequence pipeline using what's called the reflow runtime. So reflow is kind of like SnakeMake or Nextflow or any of those pipeline management systems, but it plays very nicely with AWS. And basically, given the user inputs for a given sample, reflow outputs a run file that reflow can execute. And then given a bunch of reflow run files, reflow can output a single batch run to execute. So basically all you do is, given today's sequences, let me just generate run files for all of them, which is a simple command. And then let me now generate a batch file to execute them. And then I can just do reflow, run, and then my batch file, and it just executes. So the importance is that this is modular. So there's not a complete consensus on what tools are best practice for each purpose. And with this, you can now pick between, maybe I want to use Minimap2 versus maybe someone else wants to use BWA. There's different tool choices that the user can easily just switch between very easily. It's scalable. So reflow has an AWS EC2 cluster manager that distributes jobs. And importantly, regardless of how many samples you have, essentially, the usage is identical. And reflow will automatically handle spinning up sufficient EC2 instances for you. And then reflow will execute the pipelines using pre-built Docker containers that I maintain. So then the user can pick between these tools without having to worry about how to set them up. So I'm just going to kind of maybe quickly end up with some results of some preliminary results. So here's a plot from some preliminary benchmarking that we've done that shows the total runtime as a function of the total number of samples that were analyzed. So on the horizontal axis, I have 1 sample versus 10 samples versus 100,000, 10,000, and I have the total runtime. And as can be seen in the current version of it, things seem to be working pretty nicely up until roughly 1,000 samples. And consistently, my total runtime is under 10 minutes, which is amazing. Unfortunately, there seems to be a hiccup that we're trying to investigate where once we hit 10,000 samples, the overall runtime blew up. And we're not really sure why this is. It seems to be something to do with the job scheduler that starts getting clogged up at this point. So we have to investigate. But fortunately for us, so NovaSeq is one of the Illumina sequencing machines, and it's kind of like the big, hefty one that can sequence a bunch of samples. And in general, all of our experiments have been right around 600 to 1,200-ish samples that we can execute in a single NovaSeq run. And in NovaSeq, the machine itself takes roughly 24 hours to generate the sequences anyways. So given that it seems like in its current form, we can scale such that a single NovaSeq run is handled basically appropriately, we seem to be in a good spot that we can deal with for now while we try to debug this big jump. So I don't want to go too much into the specifics, but you can check out the GitHub repo if you're interested in seeing how it works. But some concluding thoughts, some specific future directions. The obvious one is we want to fix why this jump in runtime happens from 1,000 sequences to 10,000. We want to implement more tools as options in the pipeline so that it can just be a one-stop shop for anyone to have any non-expertise and just kind of pick and choose what you want to use. We have a very useful command line interface, but we have a really bad GUI that I just kind of slapped together in a few hours. So we want to have a more user-friendly mechanism for non-command line execution. And largely because a lot of the people that are doing these sequencing experiments don't have very much computational expertise. So it really needs to be like as easy as just typing in your AWS credentials, click the button and have it run for you. And right now, because it uses reflow, it's very specific to AWS, but in the future, we want to try to generalize it to non-AWS cloud platforms as well. So some acknowledgements. I want to acknowledge my co-authors and the other folks that have helped me with this project. And yeah, I'll leave the rest of the time for any questions that people might have.
Oh, thanks for the talk, Niema. That was really, I feel like that packed a huge amount of different domains of knowledge into a really effective presentation.
Thank you, thank you.
So just a reminder, if you have any questions, then drop them in the chat. In the meantime, we have a few that have collected so far. First question is, could you share any strategies that you've taken to pay attention to the amount of money spent and like tracking the budget for these instances that get spun up?
Yeah, that's a great question. So actually, so the paper is being written right now, but I have some results from that. Let me pull it up real quick. Basically, what it's coming down to is even in that massive spike that we saw at the 10,000 sample portion, we were hitting, I think, 11 cents per sample in terms of compute. And for the ones that we didn't have that massive spike, we were hitting, I think, like 4 cents, no, 0.4 cents per sample. And for context, just the wet lab side of generating these sequences is roughly, I want to say like $100-ish per sample. Here, let me post a link in the chat that I'll also share the screen of. Here's just, as we're drafting the paper, some basic visualizations and kind of computations. Yeah, so we're seeing, and I have it organized in a table as well, but basically consistently we're getting like 0.4 cents per sample. Oh, this is time, sorry. Here we go. So yeah, so roughly, wait, is this correct? Yeah, so this is 0.04 for 10 samples, 0.4. This is, now we're in dollars. This is in US dollars. So 0.4 for 100 samples. So yeah, $0.04, not cents. Sorry, I'm all over the place. So it's very cheap, especially it pales in comparison to the cost of just generating the sequences biologically in the first place.
Cool. And we'll record that link that you just dropped in the chat and put it in a second draft of the proceedings so that people will have access to that in the future.
Oh, perfect. Thank you.
We have a question in the chat. Can you explain your reasoning for choosing reflow? Have you considered other workflow systems that are also focused on bioinformatics?
Yeah, that's a great question. So our main reasoning for using reflow, so we considered SnakeMake and Nextflow as well. The main importance for us was just we needed it to be as easy and kind of hands-free for someone to just enter their credentials and then pick the file they want to run and have everything just scale automatically. So I couldn't find a simple way to do that for SnakeMake and for Nextflow. It could also just be my limited experience with them. But with reflow, basically, you enter your AWS access ID and your secret key and just run the configuration, which is very user-friendly. And then everything after that point just scales automatically. So it was mainly just to minimize how much parameterization the user would have to do. But I think when we try to generalize away from just AWS specific, I think we will have to leverage Nextflow or reflow, and hopefully, or Nextflow or SnakeMake. But hopefully at that point we can come up with an intuitive GUI that'll help the user kind of navigate any of those technical things.
Cool. And on that note, we have another question. How hard do you think it will be to move from AWS to another provider? Do you think cloud vendor lock-in will be a problem?
Yeah, I do think it'll be a problem for buy-in, at least at the beginning stages. So I know that in a lot of public health labs, there's some kind of red tape in terms of what services they're allowed to use, just either because of funding restrictions or other kind of rules. So for now, I'm only kind of focused on the San Diego efforts that are going on right now, which we're leveraging AWS already. So that was one of the reasons why I chose to go with this. But yeah, for more broad adoption, I really need to find a way to untether it from being AWS specific, maybe by having different versions of it or something that helps with that, but keeping it user friendly.
Cool. We have another question in the chat. Since your operation is time sensitive, I presume you avoid interruptible or spot instances. Could you comment on this?
Yeah, absolutely. Yeah, great insight. So fortunately, reflow supports spot instances if you're doing something that's not time critical. So for our analyses and for these benchmarks that I got with the $0.004, so 0.4 cents per sample, those numbers were not using spot instances. So it seems to be quite affordable, especially compared to the cost of just generating the sequences biologically, even if you use the on-demand instances. But if you, I can imagine like maybe in the future, once you have a repertoire of a bunch of sequences that maybe you want to reanalyze if some new tools come out in the future, I think spot instances would be pretty helpful for that if you aren't as time sensitive for reanalysis, if that kind of makes sense. But yeah, for our purposes, we avoid them.
And just to clarify for viewers who aren't familiar with the term, spot instances are virtual machines in the cloud that are preemptible in case a different customer has a more important workload to do. So they cost an order of magnitude less than you usually pay, but they have this caveat that they are very only as available. So thank you for that question and that answer. We have another question. Let's see, what's the next one? There are so many good questions. Have you encountered any challenges in taking pre-existing tools and adapting them to running in the cloud on virtual machines, or has it been a smooth process?
It's been fairly smooth for the tools that I've tried so far. So I think the main one that I've struggled, so fortunately, most of the kind of high performance bioinformatics tools that are used are written in something like C or C++. One trouble that I've run into in the past is some tools are written in Java, and with reflow, I have to specify how much memory I want my instances to have. And then just the Java runtime environment, like just JVM sometimes needs, I guess you could try to play with how much memory you allocate to it upfront, but it's not as trivial or like hands-free as just running a command that happens to always work no matter what your scalability is. So yeah, so for the most part, no, it's been fairly simple, but there have been a couple weird cases. I'll also say maybe just on that note, so I'm using Alpine as my base image in the Docker containers because it's extremely minimal and I want to keep the runtime short in terms of it has to pull the images with the tools. But Alpine is super minimal and the C libraries that are on it don't necessarily always translate to, like, I think it uses muscle versus it doesn't use glibc. So there was like weird issues that I ran into getting stuff to compile, but once you get it working, I don't know, it's like a couple hours of kind of tinkering with it and it starts working.
Cool. And so I think the last question that we have before a break, CloudBank is really interested in figuring out how to help teach debugging skills. And to this effect, we're wondering if you've engaged at all with an AWS solutions architect to dig into that runtime jump that you see. Yeah.
Yeah, we haven't asked them yet. I think that would be hopefully a next step. I guess my concern is I don't know if it's an AWS side thing or if it's a kind of local job manager thing that's going on under the hood. So our main bottlenecks were our AWS accounts had, so basically Reflow will try to distribute your jobs to a number of EC2 instances that has enough CPUs based on how many jobs you're running and how many CPUs each one needs. For our purposes, we just use one CPU per job just because each individual sample, even single threaded, is fast. But really quickly, we ran into an issue where our vCPU limit on our account was too low. I think it was like 300 by default, whereas we needed it to be like 300,000 or 200,000 or whatever. So there was a couple of marketing bottlenecks in terms of just like requesting expansions to those limits, but those are pretty easy to get through. But yeah, I think hopefully I can get in touch with people that are better with AWS than I am to see what's causing this jump.
Cool. Well, just as a cloud education specialist, I think that I can say I'm very interested to hear back about, what the process that you followed ended up being for solving that kind of bottleneck, because this is the kind of thing that, like, I think our community of practice can really benefit from, just learning about these kinds of hiccups and learning how they've been overcome.
Yeah, absolutely. I can touch back afterwards. I mean, fortunately, the simple workaround is the jump was like an order of magnitude more runtime, almost two, versus, you could just split up your data set into 10 pieces and run them individually. But then we get back into the whole issue of now it's getting repeat like manual effort anyway. But yeah, either way, but I can loop back in once I know more.
Yeah, maybe we can just write a Perl script to do that for us.
Yeah, exactly.
Anyway, thank you so much for the presentation, Niema. So we will now pause for a break and reconvene at 12:30 for a vendor presentation from Microsoft, where we will learn about the state of research on Azure today. I'll see you all in 10 minutes.